Excellent. So first of all, again, thanks very much to Sophie and Desi for the invitation to present in this Chicken Journal Club. I'm very impressed with your organization, Twitter and all the YouTube and everything. It's really good. And I wasn't aware of some of those upcoming talks, so it's really nice to know about them. Right, so the title of my talk is Campylobacter and the Chicken Microbiome. I'm Ozan Gundogdu. I'm from the London School of Hygiene and Tropical Medicine. And today, really, I want to give you a sort of overview of the work that we've been doing in terms of chicken microbiome work, but also how essentially we came here, which was essentially from our historical work on Campylobacter. So a bit of background. Historically, we studied the physiology and the pathogenesis of Campylobacter and other foodborne pathogens. So this is really kind of my historical work at the school for nearly 17, 18 years now. And that involved a lot of basic characterization of genes that were involved in essentially survival of the bacteria. And I would definitely say in the last sort of 5-6 years, this has moved on to trying to incorporate our knowledge base from this sort of basic microbiology, if you like, pathogenesis and physiology, and translating that, moving that towards the real world. Because I think there has been a bit of a discourse in terms of Campylobacter between those two things. And my background is molecular biology, but also computational science. So I try to always incorporate omics-based technologies into the research. And the sort of research area is covered in two main themes, as I was just mentioning. So we kind of have this basic microbiology research where we look at virulence factors, pathogenesis, survival, properties of the bacteria, and how can we utilize this knowledge base in terms of translational research, devising intervention strategies, looking at things like management practices, and obviously the forever link with Campylobacter and chickens. And these are the members of my group. They're all ex-MSc students. Currently, Zara Omole is working on the type 6 secretion system. She's just started. Janie Liao is now a postdoc who finished her PhD successfully on the type 6. Gemma Hong is working on the unfolded protein response, cellular-based research, looking at how Campylobacter actually causes diarrhea. And Katie Davies is working on outer membrane vesicles, which, for those of you who do not know, they are essentially phospholipid blebbing off membranes from the bacteria that can contain interesting components of DNA and protein and proteins of interest. So this is basically the team at the LSHTM. So a little bit about Campylobacter. I'm not sure how many of you work with Campylobacter. It's gram-negative, but I guess one of its main sort of interesting sort of characteristics is that it's microaerophilic. And that's always been an interesting point, because if it's meant to grow at 5 to 10% oxygen ideally, and in the sort of atmosphere we have approximately 19.1, 19.2% oxygen, it's omnipresent within the environment. And how is it able to do this? Clearly, it must have good genetic regulatory mechanisms for the bacteria, for it to allow it to survive in these non-ideal conditions. And then this optimum growth temperature of 37 to 42, which obviously is humans, and then the avian body temperature of 42. And so the main sort of disease causing species are Campylobacter jejuni, followed by C. coli. So historically, Campylobacter is not really considered a model organisms for two main reasons. And that is that we haven't really had, I would say, good, convenient animal models. And also the difficulties historically in terms of growing the bacteria. And so this has been, it's known when you talk to people and you say you're working on Campylobacter, they're like, why would you do this to yourself? It's quite a pedantic bacteria to grow, and sometimes you don't understand why it's not growing as well as you'd think. And so this was one of the main reasons why, because of its sort of health issues and socioeconomic issues, it was one of the first bacteria to actually be sequenced. 1999, 2000, Julian Parkhill and various people at the Sanger Institute sequenced this, and they found a sort of a number of key attributes of the bacteria, key loci involved in lipooligosaccharide, external capsule, which at the time was debated. You had people in conferences standing up saying there's no capsule. And one of the people, Andrei Karlyshev, actually proved just before the genome sequence came out that actually with an Alcian blue staining, it does have a capsule. And you had people that had worked on the bacteria obviously for many a year that were still saying, no, it's not there. There's no capsule. But when the genome came out, it was 100% clear that it does have the gene sets, the loci for the capsule. And it also has some interesting glycosylation systems, O-link glycosylation on the flagella, allowing evasion of the immune system, and N-link glycosylation, which is very interesting because historically, I think this was one of the first bacteria to have N-link glycosylation, if not the first. And it's glycosylation of periplasmic and outer membrane proteins, allowing a multitude of functions from effective folding and heat resistance and various other functions. And so these were interesting findings. Interestingly, no type 3 secretion system was identified, which is quite classic, enteric bacteria not having that. But from our work, more recently in the last 5 years, there's been a lot of strains that have had the type 6 secretion system. And so if you look here, we actually updated the genome in 2007. And even within that 7-year gap, there was almost 20% of the genes were updated. So from a genome of 1,654 to have 20% of the genomes updated, we believe was a worthwhile exercise for the sort of research community to have the latest characterized genome file. In terms of disease profile, causes gastroenteritis, usually self-limiting, and there is this difference in terms of disease phenotype, if you like, from high resource countries to low resource countries, where in high resource countries it's typically a fever, bloody diarrhea, and whereas in low resource countries it's typically associated with watery diarrhea. And one of the potential explanations for this is the regularity of exposure in low resource countries at a younger age. And obviously it can cause post-infectious complications such as Guillain-Barré syndrome, which is the sort of most famous one, where there's molecular mimicry of the outer gangliosides caused by the lipooligosaccharide of the bacteria. And so in terms of numbers, generally it's really widely underreported. So the numbers that you typically see, are a sort of a 1 in 10 roughly of actually what's the current situation. So in the UK, 65,000 reported, but this is believed to be more in the line of 700,000, and it's simply because most people that contract Campylobacter don't go to the GP to get actual testing, and so there's huge health problems and also economic and social problems. And so then obviously this is a chicken club talk, so there's obviously a link to chicken. We're not going to spend any more time really on Campylobacter in that sense, but it's to give a background of how we ended up coming into this field. And we know that obviously the main route of infection to humans is via chicken, via poultry, via handling of chicken. And so this was an early figure from Young et al. And it's quite interesting because even back then, there was a hypothesis that actually, Campylobacter is in the avians. Is it a commensal? That's been a great discussion point over the years. I would say that my personal opinion and various other people that I've spoken to, in the last 5 years, we've kind of veered away a little bit from using that word commensal. And there's been a few studies from various people that have actually said, if you actually stress the chickens, you will actually get a sort of disease phenotype quite similar to humans. And so that is an interesting discussion point. And the numbers are so high in the poultry, in avians, that one of the hypotheses that actually came out is that the mucus layer is actually just not allowing that bacteria to adhere and invade for the disease progression to occur, whereas in humans the mucus is different, and adhesion and invasion does occur. So that's still work really in progress. We're not 100% sure why that is the case. And so when I started working on Campylobacter, everybody knew about Salmonella, for example, but just talking to people in general, they weren't aware of what Campylobacter was. And definitely, I would say in the last sort of at least in the last 5 years, the UK government has made a major push to really educate people about Campylobacter. Just people are much more aware when you talk to them about Campylobacter of what it is. And there are all these advertisements saying don't wash raw chicken. There was a bit of a hoo-ha when members of the parliament were on TV saying just get your chicken and put it straight in the oven, and without a full explanation that this is because there's bacteria on the surface of the chicken. So you just want to limit the spread of this bacteria to your kitchen utensils and your vegetables that are lying on the side. And so all kinds of statistics came out at this stage about which supermarkets have the most Campylobacter on their chickens. So at this stage, we kind of were getting interested in where maybe we can utilize all our knowledge that we have in terms of laboratory pathogenesis physiology and see actually why is Campylobacter within the chicken in such high numbers that's not causing disease that we're seeing more regularity in humans, if we put it that way. And so, but simultaneously, coming from the chicken side of things, we know, that, and all of you know this, that chickens are a key source of proteins. And, but, there's two sides to this, that you obviously want to reduce the bacteria that are potentially harmful to humans, but you're obviously looking at the same time in terms of sustainable poultry practices and increasing the sort of production. It's a balancing act, because obviously companies will want to maximize their output, but simultaneously reducing bacteria that are potentially harmful, not only to humans but to the chickens themselves potentially. And so research supporting the overall health of the chickens. Ideally, we want to translate this to intervention strategies again. To potentially lower the burden of harmful bacteria. And so when we started this workout in 2015, this was performed really, what we wanted to do is to not just do this as an academic study. So we really linked with two key players that have not only been, this project would not have run without them, but also in terms of funding. And it's been really refreshing that industry, in this instance, Moypark, which I think now is the second biggest poultry supplier in the UK, has funded these projects, really, essentially because the Food Standard Agency in the UK is obviously wanting more action from companies to help with investment to lower this. And they've been fantastic in terms of their funding. And AFBI is the Agri-Food Biosciences Institute, and they are a sort of public government body under DAERA, Department of Agriculture, and one of their bodies based in Northern Ireland. And so these have been sort of key players in what we've been doing here. So our first study was really, one of the questions that we kind of were asking is, we were not sure, and this is obviously something that we're always learning more about, is how is the day-to-day microbial population structure changing within the chicken gut microbiome? Okay, so this is obviously linked with our intention to try and study Campylobacter, because what we actually sort of envisaged is, if we actually learn more about the chicken gut microbiome on a day-to-day level, then that may allow us to infer new insights into how, why, and when Campylobacter comes into chickens. And so we'd seen a number of studies at this time, even in the last few years, that had looked at this, but nothing in a day-by-day level with sufficient replicates really to try and tease out this information. And we also wanted to look at, for example, key things like what are the driving factors for the change in the microbial population structure over time within chickens? For example, competitive drivers or environmental drivers, and to really investigate these with statistical analyses. And again, there was some dogma at that time that Campylobacter, typically you start observing it within a 2-week period within the chicken life cycle. And so we wanted to test when we observe this, and is this the case as in our studies. So this particular study, we followed the chickens from day 3 to day 35, and every single day we had 12 different chickens that we basically were able to sacrifice and isolate the genomic DNA from the seeker, and simultaneously look at things like chicken weight and feed conversion ratios. So in terms of sort of pictorially, what that means is this kind of setup. So we have essentially 12 pens, and in each of these pens there is 33 chickens, and every single day, for example, the first day, day 3, we randomly took a chicken and managed to isolate the genomic DNA from the seeker, and so forth, day 4 and day 5. So really going through this in a sequential manner. Now, there is a problem with this in terms of study design, because it's not really a longitudinal study. And the reason it's not longitudinal is because every single day we're taking out one of these birds and we're not putting them back in. And so this was a decision that we had to make early on to say, are we going to try and look at the same birds over time? So one of the ways that we could envisage doing this is via cloacal swabs. That's potentially also a rather invasive method for the bird, and you wouldn't really want to do that every single day. That's an important point to make. And also, it's not really representative of a specific organ of the microbiome. It's more of a sort of a generic gastrointestinal picture. And also, when you do that, there may also be changes by the time you manage to take that sampling point. So what we decided for this particular experiment, let's sacrifice the birds and take the genomic DNA directly from the seeker, and really try with the replicate number, really try to get a good picture of what's happening in terms of microbial population structure, and to discretize potential competitive or environmental factors that may be having a role in some of these changes over time. And so the methodology is pretty standard now. This was performed 5 years ago, and it was traditional 16S, and most of you will know this already. So the results, so the first thing that we did was we looked at microbial diversity with classic environmental metrics such as richness and Shannon. And so the first thing to note is, yes, there is an increasing microbial diversity over time, but for this particular experiment, because of the metadata that we were collecting in terms of bird weight, feed conversion ratio, and a few others, these were grouped into different sort of weeks, if you like. And so the data is presented in those blocks. And so no surprise here that richness and Shannon show increasing microbial diversity over time. And one of the things we were keen to look at was what's the sort of level of variation between the pens, and I guess somewhat in a good way, there wasn't much actual variation between the pens. So we found very little differences between those. So in terms of biological replicates sampling points, we were quite happy that changes that we will observe are not going to be due to simply having chickens in different pens in this setup. And so when we look at microbial diversity over time, what we have here is the days on the x-axis, and Shannon microbial diversity on the y-axis. So we see a rapidly increasing microbial diversity and then a plateauing effect, which is expected. So each of these time points should have 12 replicates representing the 12 different pens. Now, interestingly, the triangles are where we observe Campylobacter. Okay, so there's a couple of things to note here. Firstly, that again, we observe Campylobacter in this day 16, which is incredible, because that's what the sort of our discussions over the years with people that have done this kind of experiments, that is when most people have noted observation of Campylobacter at the sort of 2-week time point. Now, one of the interesting things is that one of the chicks had this at day 3. Now, these animals were brought into this pen, so there may have been a contamination at that point. We don't think that Campylobacter at that time point is typical, although there is, there are some discussion points about that. Some people do believe that you can have Campylobacter at that time point. And the other thing to note about this study, and it was, this is always, I guess, one of the risks that when you do this sort of experimental setup where these were performed in the AFB sort of animal farm facility setup, which is a dedicated farm that essentially is an experimental setup that mimics a farm where we can modify experimental settings. The risk you kind of have with it is that there was no specific infection from Campylobacter. We did not infect Campylobacter to these chickens to say let's answer this research question. We took the risk to say let's try and see what is happening naturally, and the problem with that is you really don't know what's going to happen. The interesting thing that happened in this particular study in terms of Campylobacter is that 25% of the samples did have Campylobacter at day 16. However, we did not observe Campylobacter after that time point. And this was a bit of a surprise because we would have expected there to have been some spread. Although, as you know from the experimental setup, we did, if these birds did have Campylobacter and we're not sure where they came from, maybe the taking that bird out and sacrificing it, it did minimize the spread from typically, for example, feces to the other birds. But it does beg the question as to how, why, and when did the Campylobacter actually come in and be detected at that specific time point. This was the sort of location of the pens where we observed the Campylobacter. And it is interesting to note that 2 of them were side by side. Now, the sort of Shannon and richness alpha diversity metrics was followed up with some beta diversity metrics, looking at comparisons between the different groups. And here we have some sort of classic PCA plots looking at Bray-Curtis. So in this instance, we're looking at differences in terms of number. So what that means is bacteria X might be present in this sample 5 times, bacteria X might be present here 500 times. And so the difference that you're seeing in terms of Bray-Curtis is in terms of numbers. So what you're seeing is that in the early time points, there's a lot of variation in terms of this. Maybe the similar bacteria are there, but they're varying a lot in terms of their actual numbers, in terms of the actual counts that we detected. And then in terms of UniFrac, looking at phylogeny-based method, more essentially the different types of bacteria. Again, a somewhat of a convergence. If you squint, you may be able to see that. And then finally, the weighted UniFrac, which is the combination of both. And so we followed this up with a number of different studies. So things like local contributions to beta diversity. Again, essentially how far away from the average are the samples, so at the earlier time points. Process. So we I've just got a sign up saying my internet connection is unstable. I hope you can still all see me and hear me. So at this stage of the analysis, one of the things we were looking at was how can we further study this to potentially look at impact of competitive or environmental factors. And so we used this this sort of algorithm where we actually could try to tease out sort of dispersion and in terms of what is actually happening here. Is there essentially like a hand of God effect where there's a bit of control in terms of what bacteria are there, or is it more random? And so the sort of analysis looks at what's called NRI, net relatedness index, and NTI, net taxon index. And for both of these instances, a negative value, so the more lower the value on the y-axis, implies stochasticity over dispersion, and this implies essentially less environmental, more competitive factors having a role to play. Whereas a positive value for both of these implies more environmental impact, essentially clustering deterministic. So what does this mean? What it means, based on our experimental model, is that at the earlier time points, competitive factors are really driving the microbial population structure. And that kind of makes sense, because there's obviously a lack of potential food, there's a space topic. And so at that earlier time point where the microbial diversity really is growing rapidly, that's driving that microbial population structure. But what we observed was this somewhat dramatic shift, where at sort of between sort of a day 11 and 20, this shift to environmental factors having a key role in terms of determining microbial population structure. And this environmental factor doesn't necessarily mean, just as we know it, the external environment. Because the microbiome of a chicken is a constrained environment, it could actually represent the chicken itself acting as an environmental factor. And obviously, the life cycle of the chicken comes into play here, in terms of the chicken is growing, becoming more healthier, and so there is this observable shift in terms of microbial population structure. So when we overlaid pretty much most of these different analyses that we produced, so what we observed is that there is this, not only this sort of rapidly increasing bacterial microbial diversity, the local contributions to beta diversity at this sort of section leveling out, and then this the impact of environmental factors. We kind of said, okay, maybe we can this is almost like a window of opportunity where the microbial population structure is a little bit trying to stabilize, but it's at that key point where it hasn't yet stabilized. And maybe something that gives an opportunity for something like Campylobacter to be detected classically at sort of a 2-week mark. And so the analogy we use is, if you have a hybrid car, you may nicely use electric, but if and it's running fine, but you run out of electricity, you have to go to the petrol, and that's fine. But it's that intersection of jumping between the electric to the petrol, there may be a problem. I'm not sure how the cars exactly do that, but that conversion may be a point where there could be issues. Distinct, and so we obviously find the bird weight increasing over time, and just expected measures, if you like. And so the summary of this study is, we found a rapidly increasing microbial diversity up to day 12, with variation observed both in terms of genre and abundance before the stabilization of the microbial diversity after day 20. And in particular, we identified a shift from competitive to environmental drivers, specifically between day 12 and 20, potentially creating this window of opportunity where Campylobacter can appear. Campylobacter again was identified at day 16, and when we look at the microbiome data, we can actually see the greatest metabolic changes in terms of microbial profiles were observed 1 to 2 days before identifying Campylobacter. This microbial variation over time is actually also in this experiment. We didn't go into it too much, but the diet actually did have a major difference, because these birds are typically grown up to day 35. There are different programs where birds are grown up to 55 and 70 odd days. And the feed changes. So you have like a starter, a grower, and a finisher feed, depending on, for example, protein ration or energy ration within the feed, depending on the different time point or life cycle of the bird. And changing the feed has a huge impact on the microbial population structure. And this is something that we observed. And so I guess the sort of the uniqueness of this study is that we managed to sample every single day in a sort of natural setting, or as most natural as you could get. And so this led to interesting discussions with the government and AFBI partner and Moipark, as in terms of intervention strategies, because if Campylobacter, a lot of companies use intervention methods at a late time point, maybe even after day 30, definitely after day 25. And so there was a discussion point actually that if you're going to use supplements potentially to lower Campylobacter or to affect microbial population structure, based on this study, actually, it may be prudent to apply some of those interventions at an earlier time point. And this is one of the sort of end statements that came out for this. So this was published in 2018, and from this study, because obviously we saw a huge impact of environmental factors, we led to the second study. And Desi, I'm just conscious of time. How much are we doing? Okay for time?
Yeah, we can talk. You can talk maybe about until 45, and then we can have 50 minutes questions.
Okay, excellent. All right, that sounds about right. So this initial study led to the second study, and now we said, okay, we've observed this environmental impact on bacterial population structure. Let's now try and look at this in a real world scenario, in an actual farm. And so in this instance, we obviously wanted to really measure an amazing amount of extrinsic parameters that are happening in different industrial farm setups, to really try to compare microbial population structure dynamics, and potentially, again, Campylobacter is always there, as in what are the effects of these parameters, not just on performance, but also Campylobacter. And so we looked at 3 different industrial farm setups. So one of them was classified as a normal farm. The second was their high welfare, and one of the major differences with the high welfare compared to the normal is an increase, is a decreased stocking density. So essentially the birds have more space. And then the third one was the same high welfare farm, but this time with omega-3, because there's a lot of anecdotal data on the addition of omega-3 improving the performance of the birds. And so in this particular instance, we focused on day 7 and 30 because we were more interested almost in that, based on our previous study where we had that window of opportunity, what's happening before and what's happening after, because there is this potential turbulence in the midpoint. And so really the strength of the study, I'm not sure you will be able to see this, but it's a huge amount of measurements that we took from different external parameters in different farms. And this really was the strength of the study. And this is only half of them. And so some of the kind of things that we've observed, again, looking at alpha metrics, looking at microbial diversity. This time you'll notice that basically we have day 7, the free farms, and day 30, the free farms. So those are the comparisons we're doing. And again, looking at environmental impact, which followed similar patterns to what we had observed previously. But some of the things that, for example, we did was, what are the actual microbial changes that are actually occurring, not just between the days, but actually between the farms. And this is not a trivial thing to do. And to do this, we actually used, in this instance, for example, 4 different metrics. So we used subset analysis, DEC2 analysis, heat tree analysis, and MINT analysis. So all really trying to go with a consensus. And I think that's one of the things that we've learned from doing our microbiome studies, is not to just rely on one tool or one statistical method, to really try and apply a few different and get a consensus feel for the data. We kind of, this is an example of our MINT study, and so MINT is quite used heavily recently. It gives a sort of overall discriminatory power, considering all sources of variations from the study in terms of production system and temporal scales in terms of days. And so we can overlay the phylogeny here, and you can see a lot of the different external parameter metrics that we actually took into account, how they overlay the phylogeny with this. So for example, the classic one here is, this side of the graph is day 30, for example. And if you look at treatment and you match that to what's on the graph here, O is orange, O is the, N is the green normal. So this is the normal set of data. The blue is the high welfare, and the orange, for example, is the omega high welfare. And it's really interesting that actually they do cluster together. So all of the omega high welfare did cluster together, all of the normal and all of the high welfare. And then obviously we look at Campylobacter, and really somewhat surprisingly, we only really observed Campylobacter in the normal data set at day 30. The day 30 was expected. We didn't expect to see it at day 7, but we did observe it only in the normal experimental setup. So because of all of this sort of metadata that we have, what we wanted to do is actually say, okay, let's try and look at this further. So obviously we do things like permanova, and that shows the extent of influence on the microbiome structure in terms of variability. But what about in terms of sort of directionality? So we wanted to obtain directions if there was an increase or decrease in parameters caused by an increase or decrease in the proportion of bacteria in the microbiome. And to do that, we performed subset regressions on the one-dimensional data sets, such as alpha diversity, taking the beta diversity and the local contributions of beta diversity. So, here's an example of some of this. So some of these are yellow, and that's because they're dummified, which means we don't have a measurable value for them. They're either present or not. So for example, just as one example, placement per birds, that's essentially stocking density. If you increase that, NTI is reduced. What that means, NTI was the environmental impact. So increasing the amount of birds in a confined space, you actually have a reduced impact externally to impact the microbial population structure. And this is interesting because this also matches production system N, which has the highest placement per bird, the stocking density. So this is an example of the kind of things that we can do to tease out the impact of these different extrinsic parameter metrics on microbial diversity, environmental factors, and how different things are from the norm. Again, looking at unifrac numbers, Bray-Curtis numbers, unifrac, phylogeny, different bacteria, and the combination of both, weighted unifrac. So to summarize the study, this kind of figure really encapsulates everything in this particular study. So we obviously wanted to look at which parameters are important for performance and pathogen reduction. That was the overall sort of goal of the study. And we had these 3 different production systems, and production system H, W, and O, again, the only distinction between them is that one of them had omega added to it. We managed to identify specific bacteria that were different in the actual different experimental setups. And then there were some key messages that came out. So for example, again, going back to the example that I just highlighted, increasing the stocking density, so increasing the amount of birds within an area, does seem to signify that external factors, environmental drivers, had a reduced effect on influencing microbial population structure. Omega-3, for example, did increase the weight gain of the chickens. And in terms of Campylobacter presence, interestingly, the increase in the presence of Campylobacter did seem to be linked with, for example, increase in microbial diversity, which is an interesting concept. And then there's a whole area, which for time-wise, I haven't even managed to go into, is we've linked energy percentage of ration, protein percentage of ration with specific bacteria, really looking at the effect potentially of the role of the diet in terms potentially of bacteria that can break down short-chain fatty acids, which has been highlighted quite recently as important in terms of the chicken microbiome. So to summarize, Farm N or System N significantly, it was dissimilar compared to high welfare and omega. And the example of the placement per birds led to a decrease in environmental pressure. influencing microbial community structure. Again, specific bacteria were identified within each of the industrial production systems. And again, we link this to energy metabolism, amino acid, nucleotide, and short-chain fatty acid utilization. And Campylobacter was identified in specific production systems, and then the impact of omega. And so in conclusion, our findings demonstrate this relative role of different production system parameters in shaping the bacterial community's impact on the chicken microbiome. And really, it is clear that parameters between production systems, whether they are constant or variable, have a key impact on microbial diversity, which subsequently influences feed breakdown and hence instigates competitive exclusion of certain genera. And future studies will have to look into this further, specifically in terms of the diet and the constituent parts of the diet and the impact on the microbiome. And this was published late last year. And if you're interested, I would recommend definitely go and read that further. So in summary, I've gone a bit over, apologies for that. Campylobacter is not going away. And for me personally, I think we need a holistic approach. So we obviously need molecular microbiology and the bioinformatics techniques, but we need these go hand in hand with things like good education, just educating people to cook poultry properly, for example, and obviously better government policies. I do feel when for example, I visited the Moy Park facilities, it's incredible at the level of security that they have to minimize Campylobacter infection. It's like going into a NASA space station. But I do feel, you have to question whether this is happening in many other parts of the world, for example. And vaccine development, well, that's an interesting one because there's obviously huge economic questions about that. Is this economically viable for an animal that you're potentially going to kill in 35 days? So there's obviously huge cost considerations for that. So this is a sort of an overview of different collaborators, but specifically for this project, really there's sort of 3 teams that have been critical in this. First of all, the AFBI team led by Nikolai Korchioniboshi. Many of you will know organized the previous CHRO 2019 Belfast meeting. He has been really influential in this project. The Moy Park team, particularly Aaron McKenna, Anne Richmond, and Ursula Lavery, who have shown, they were also at CHRO 2019, and really it is a great model for industry to contribute towards academic research. And also the team at the University of Glasgow, Umar Ijaz and William Sloan, who have been instrumental in the bioinformatics analyses and applying different tools to analyze the data. So that's the talk. Thank you very much all for listening. I'm happy to try and answer your questions.